The human DNA is about 3.3 into 10 to the power 9 base pairs. Both of them, they carry a positive charge. So therefore, the histone is entirely positively charged. Because at the end of the day, we know that proteins are nothing but chains of amino acids. What is the name of that linker protein? It is a H1 protein. Hello everyone, a warm welcome to this session for class 12 CBSE biology. I'm Dr. Divya, biology faculty. So in the previous session, we had learned about the DNA, the structure of the DNA and what are the salient features of DNA. So in this session, we'll learn about the coiling of the DNA, the packaging of the DNA, and we'll learn about the search for the genetic material. So I told you in the previous session that the DNA, they are very lengthy. Say for example, the human DNA is about 3.3 into 10 to the power 9 base pairs. So inside a cell, so we are multicellular in nature, right? We have many number of cells, and in each of these cells, there are nucleus that is present. Inside the nucleus, chromosome is present. In the chromosome, what is that? It is nothing but threads of DNA which are tightly coiled. Now that coiled like structure that you see, the threads of DNA, it is about 3.3 into 10 to the power 9 base pair. It's quite fascinating, right? How can they coil and sit inside one tiny small nucleus? How it happens, we will look into. So that is about packaging of DNA helix. So take the distance between the two consecutive base pairs as 0.34 nanometer. So if the length of the, say we have the DNA strands, right? Adenine, guanine, cytosine, thymine. So between these itself, there is a distance of 0.34 nanometer. So this is into how many times? 0.34 into 10 to the power 9 meters. In meters, we write it as 0.34 into 10 to the power 9 meters because we can't give a very big number. If the length of the DNA double helix in a typical mammalian cell is calculated simply by multiplying the total number of base pair with distance between two consecutive base pair, it will be about 6.6 into 10 to the power 9 base pair into 0.34 into 10 to the power 9 meter per base pair. So when you calculate, approximately inside each nucleus of the cell, there is about 2.2 meters of DNA. Now, that 2.2 meters of DNA, how can it sit inside such a tiny nucleus? So, the length that is far greater than the dimension of a typical nucleus. So, the nucleus is about 10.6 per meter. It is more than the size of the nucleus. But still, how can they fit into? That is what we will look into. So talking about the packaging of DNA helix in prokaryotes. In prokaryotes, how do the DNA helix get packed? So in prokaryotes such as the bacteria, Escherichia coli, though they do not have a well-defined nucleus. Bacteria, they are put under prokaryotes because they do not have a true nucleus or a well-defined nucleus. What do we mean by that? It means that their genetic material is distributed as such and they are not covered by any nuclear membrane. So that is why we call the nucleus of a bacteria as not a true nucleus. So DNA being negatively charged, it is negatively charged because the backbone of the DNA is made up of what? Phosphate. You have studied that in the previous session, right? If not, you can go and have a look at it. So phosphate, right? What is it made up of? It is made up of phosphate, which is PO4 minus. It is negatively charged. So DNA being negatively charged is held with some proteins. Now, if they have to be held with some proteins, like charges, we know that like charges repel, unlike charges attract. So therefore, the protein should be what? It should be positively charged. So therefore, the protein is positively charged. The DNA in nucleoid is organized in large loops which are held by proteins. So in the same way, in prokaryotes also, the lengthy DNA that is present, distributed inside the cell as such, they are held together by proteins. So the negatively charged DNA coils around the positively charged proteins and they are held together tightly. So this is how they form a loop like this. So this is one strand, this is a protein, this is a protein. DNA is negatively charged, negative backbone, which results in the formation of a coil towards the positively charged protein. So therefore, they form loop-like structures like this. This is how it is arranged in the case of a bacteria, especially in Escherichia coli, which we have taken as the example. Next, let's see how are they packaged in eukaryotes. In eukaryotes, do they look like a loop itself or some other structure? We will look into. So in eukaryotes, the organization is much more complex when compared to the prokaryotes. There is a set of positively charged basic proteins called as histones. So here also, the DNA is negatively charged because the backbone of the DNA is made up of phosphate molecule which carries a negative charge. That phosphate molecule gets attracted towards the positively charged protein. And what is the name of the protein? It is histone. So here the DNA are tightly packed because of the presence of which protein? Positively charged histone proteins. A protein acquires charge depending upon the abundance of amino acid residues with charged side chains. So therefore, it is meant that histones contains a lot of amino acids which are positively charged. So therefore, histone is positively charged because at the end of the day, we know that proteins are nothing but chains of amino acids. You have studied about this in your class 1st PUC or class 11th. So histones are rich in the basic amino acid residues which are lysine and argine. Both lysine and argine are highly positively charged. So therefore, histone is also positively charged. It can come from MCQs. What is histone made up of? It is made up of high quantities of two important amino acids, that is lysine and arginine. So you can see here, this is the histone. So this is a histone protein. Histone protein is positively charged. Why? Because it has two important amino acids, that is lysine and arginine, in abundance that gives it a positive charge. Now this is the DNA. This is the DNA. DNA is negatively charged. Why is it negatively charged? Because its backbone is made up of a phosphate which is negatively charged. This positive and negativity attracts them together, making them to tightly coil around the histone molecules. So this is the core histone. And together we call it as the nucleosome. In eukaryotes, in prokaryotes, what did we call them? Nucleoid, right? Nucleoid, we call them as nucleoid. Together with the protein and the DNA, together we call it as a nucleoid. In the case of eukaryotes, we together call them as nucleosome. So nucleosome is the material that is present inside the nucleus. So moving further, both the amino acid residues, as I told you, they carry what? Positive charges in their side chain. What is their side chain? Remember, again, biomolecules. See how 1st PUC and 2nd PUC are connected to one another, right? What is the structure of the amino acid? They have a carboxyl group on one end and they have a carbon, alpha carbon atom, and they have a amino group on one end. And in the side chain, where did they mention this? They mentioned that the amino acid residues carry positive charges in the side chain. They have a hydrogen or hydroxyl group and they have a R. So here, this R can be arginine or it is lysine. Both of them, they carry a positive charge. So therefore, the histone is entirely positively charged. So histones are organized to form a unit of 8 molecules called as histone octamer. So is it made up of just one histone? No. There are different types of histone proteins that are present. In eukaryotes, especially in we human beings, for example, we have the histone proteins H2A, H2B, H3 and H4. So how many are there? 4. These 4, in how many numbers they are present? 2 each. So therefore it will be 8, right? 2 to 4, 5, 6, 7, 8. 8. That is 2 proteins of 2 H2A proteins, 2 H2B proteins, 2 H3 and 2 H4. Therefore 8. That is why we call it as an octamer. So this is an octamer arrangement. So say for example, we have H2A, one more H2A, then we have H2B, one more H2B. Here there is a H3, one more H3. Here we have a H4, one more H4. Okay, behind. So it is an octamer. And the negatively charged DNA, this one, the negatively charged DNA will coil around the positively charged histone. Now, only coiling is enough? No. Once it will coil, can everything coil around the same histone octamer? No. They need to have many histone octamers in which they coil. So, we'll look at that later on. So, the negatively charged DNA is wrapped around the positively charged histone octamer to form a structure which is called as the nucleosome. Now, this is what we call as the nucleosome. We have seen it in the previous slide itself, right? So, moving further, we'll talk about the nucleosome. Now, each nucleosome. What is one nucleosome made up of? It contains histone octamer around which one stretch of DNA is coiled. Next, again, one more nucleosome. It is made up of again histone octamer around which DNA is coiled. Can you see here? This is one. This is the second one. First nucleosome, second nucleosome. Here also it is an octamer wherein it is made up of 8 molecules of histone proteins. Here also it is made up of 8 molecules of histone proteins, everything. And together they form a bead. Now to make it a bead, we need a connection between one bead to another bead. Now what is that connection? That to form a connection also, it is made up of a protein. That protein, we call it as a H1 protein. That H1 is a linker protein that links between two histone octamers. That is, between the first histone octamer and the second histone octamer, there is a H1 protein. Between the second and the third histone octamer also, there is a H1 protein. Understood, right? Here also H1. Here H1. So, therefore, because they link each histone octamer, we call it as the linker protein. It's also called as the linker protein. So, all these are important for MCQs. So, nucleosome constitute the repeating unit of a structure in nucleus, which together we call it as a chromatin. Now, this together, we call it as chromatin. You have studied chromatin fibers and all that in your previous class. A typical nucleosome contains about 200 base pair of DNA helix. So, one nucleosome. So, around one nucleosome, how much DNA is coiled? 200 base pair of DNA is coiled. The nucleosome in the chromatin are seen as beads on a string. Doesn't it appear like a bead? Yes. What are they held together by? By one linker protein. What is the name of that linker protein? It is a H1 protein. Beads on a string structure where weaved under the electron microscope. So, when we see it under the electron microscope, it looks like beads. The beads on a string structure in chromatin is packaged to form chromatin fibers and are further coiled and condensed at metaphase. So, in metaphase stage, they condense further, they come and align at the center. What is it? It is nothing but chromatin fibers, and they contain these histone proteins around which the DNA is being coiled. So, the packaging of chromatin at higher levels requires additional set of proteins that are collectively referred to as non-histone chromosomal proteins. So, some additional proteins are also required if the DNA has to coil packaged tightly. Those proteins are called as non-histone chromosomal proteins. So, there is the difference between histone proteins and non-histone chromosomal proteins. So, histone proteins help the DNA to coil around to get packaged properly into a nucleus. So, non-histone proteins for complex packaging of DNA, non-histone proteins are needed. So, this is about the structure of the nucleosome. So, nucleosome, to the entire bead-like structure, we call it as the chromatin, right? So, the chromatin in a typical nucleus, some region of the chromatin. Now, this chromatin can be sometimes, or the coiling of the DNA around the histone sometimes can be loosely coiled or it can be tightly coiled. So, if it is loosely coiled, we call it as euchromatin. And if it is tightly packed, we call it as heterochromatin. Euchromatin, they take up strain very less because they are tightly coiled. So, because they are loosely coiled. And heterochromatin, they take up too much of strain because they are highly coiled. Say for example, a bead. You spread the bead and you keep it. Which one is more visible? The one that is spreaded. The chain that is spreaded or chain that is made into a coil and that is kept. The one that is coiled is more visible, right? Same way here. So that is why euchromatin takes up a light stain. Heterochromatin takes up a darker stain. And euchromatin is said to be transcriptionally active. Which is transcriptionally active? Euchromatin. This is very important for MCQ. A DNA, if it has to be transcriptionally active, meaning if the DNA has to transcribe into mRNA, then what should be there? It should be a euchromatin. Then the DNA helix or DNA should be packed loosely around the histone. If it is packed tightly around the histone, then the process of central dogma principle will not take place. And in particular, if I have to say, transcription process will not occur. So therefore, if transcription has to occur or if the DNA has to be transcriptionally active, then it should be in euchromatin form, that it should be coiled loosely around the histone. This is important for MCQ. So this is about chromatin. So this was about the session wherein we learnt about the packaging of the DNA, how exactly, what are the proteins involved in the packaging, how they form an octamer, what is euchromatin, what is heterochromatin, all that we learned, right? So in the coming session, we will learn about this search for the genetic material. That is how exactly we got to know that DNA is the genetic material that is carries information from the parent to the daughter cells, and they name the genetic material as DNA. So this is what we will study in the coming session. So we see you in the coming session.